vision research is just incredible. But I think Chris Sander just approached me that he would love to give introduction more formal. So Chris, all yours. And
great, thank you, Professor. Hello, everyone, and welcome to this week's lecture. This morning, I have the honor of introducing Professor Anand Swaroop. Professor Swaroop took his PhD in biochemistry from the Indian Institute of Science in Bangalore, India, and after completing postdoctoral training at Yale University in molecular biology and human genetics, he joined the faculty at the University of Michigan as an assistant professor in the departments of ophthalmology and visual sciences and human genetics. Dr. Swaroop was promoted to full professor in 2000 and was then recruited in 2007 by the National Institutes of Health to establish the Neurobiology-Neurodegeneration and Repair Laboratory at the National Eye Institute for advancing research in all aspects of retinal biology and disease. Professor Swaroop's lab has focused on AMD and the regulation and dysfunction of genetic and epigenetic factors in photoreceptor development, aging, and neurodegeneration, as well as the design of new therapeutics using cell, gene, and small molecules. He has trained over 500 students, interns, and fellows, and a number of his trainees hold faculty or scientist positions at institutions worldwide and in the biomedical industry. Dr. Swaroop is a leader in the field of retinal development and degeneration and has received many honors for his contributions to including the Board of Directors Award from the Foundation Fighting Blindness and the NEI Directors Award. Dr. Swaroop has published over 350 peer-reviewed articles, invited chapters, and reviews, and has several patents to his name. In addition, he's delivered over 300 invited lectures worldwide, so we're obviously in for a treat. Everyone, please welcome Professor Swaroop.
Okay, I'm going to share my screen here and okay, so first of all, I'd like to thank my brother Chris and Gresna for inviting me there via Zoom. I think my first ever Zoom talk. And so thank you all for joining, and thanks to a lot of old friends and new friends that I was able to see via Zoom and connect with. What I'm going to do is, what actually I've tried to do is prepare a different kind of talk today. I think something that I have not delivered ever. So this is first time so you are my practice targets basically today. I didn't want to give my old NRL talks or old AMD genetics talks or whatever. And I thought, I should do something different, something that is new in the lab. Part of it is published, and part of it, if I get to it, is unpublished. But there are a lot of thoughts and ideas that sort of I have been harboring for a long time. And I thought I will put these together and see how it goes. So the title really I gave is a very broad one, photoreceptor aging and degeneration. I think I'm going to start with aging. By the time sort of I finish the aging part, I think it'll be over an hour probably. So if you guys are still not asleep, I can go further. But I think what I want to mention really is that mitochondria, that sort of organelle that I ignored during my PhD, I think are turning out to be the focus for our future research program. At least will become a major part of that. So before I go any further, I want to, let me see what happens here. So before I go any further, I want to actually dedicate this talk to my PhD professor. I was in Indian Institute of Science, Bangalore, just before the lockdown and everything for biochemistry department centenary celebrations there. That was the place where I did my PhD. And my professor was Professor Ram Sharma. Can you all see my pointer? So Professor Ram Sharma, so this this was on February 21st. They had the whole day celebration, and I was one of the 2 speakers invited from here. This was the first picture that we took when he came in the morning. This was sort of in the lobby area. And sadly, this was the last picture of his life, one of the last pictures. The whole day, Professor Ram Sharma was with me, and I was sort of very fond of him, and I think he was very proud of what I was doing. And the reason I want to point this is that I did my PhD on mitochondrial oxidative phosphorylation, hydrogen peroxide generation, and things like that. But I hated it at that time. I sort of, that was the time when Tom Maniatis and everyone else was making recombinant DNA and cloning genes. And I just wanted to clone genes. I did not want to have anything to do with mitochondria. But, Professor Ram Sharma was very nice to me, and we published quite a few very good biochemistry papers from my PhD. But I couldn't wait to escape to do Drosophila genetics at Yale, and then moved over to human genetics because I'm one of those deuteronobs, and I couldn't figure out many things in flies. So Professor Ram Sharma was a little bit disappointed that I'm leaving, but he always kept on sending me his reviews papers and telling me, and I met him several times. Sadly, that day after we left him, I left him after the dinner. Apparently, within 5-6 hours, he passed away in sleep. But his wife said that he was very happy and he was delighted and proud of everything. So, it was sort of a bittersweet kind of moment for me. So I wanted to share that with some of you guys, that I'm coming back to, in some sense, my roots. And this slide was the one that I presented over there. So this was the title of the talk, and this was the slide. But of course, these 2 pictures were not part of that. So we as professors or teachers or mentors tell our students that we should focus, but I'm probably the most unfocused person around to tell everyone that they should focus. My research interests are very broad, but at least good thing is they all focus on retina. And actually, I would say much of that in retina on photoreceptors. And I just touch upon maybe a little bit on both sides of photoreceptors, but primarily focusing on photoreceptors. We look at healthy phenotypes and we look at disease phenotypes. And you can see I put aging as part of the healthy phenotypes. After all, disease is when you have to go to a doctor and say, okay, doc, I can't see or I can't breathe or whatever. But, that's not the case in healthy aging. That's what we like to call it. And the goal of a lot of the work that we are doing eventually is to design therapies for distinct disease phenotypes so that some of these can be converted to healthy phenotypes. So that's the broad goal, and use the basic science knowledge to design those treatments. So, but much of it is sort of basic, maybe a little bit translational that we try to do. So let's talk about aging. So this is one of my favorite sort of pictures here. Old wine aged to perfection. And so I'm sure living in California, and even some of us who don't live in California or West Coast, we enjoy some good old wine, which has been aged well. As human beings, whether we age well or not, it really depends, on many different environmental factors. But in almost all cases, there is a progressive decline in function and all sorts of tissue functions and cell functions. We also have a reduced response to stress conditions. And of course, another sort of a factor that comes along is the increased susceptibility to disease. So aging has always been an enigma, and people have been trying to understand, why do we age? And can we understand that from model systems and otherwise, and figure out how can we live longer. But more than living longer, I think how can we have healthy aging. So aging is a universal process. This is a picture I took from Current Opinion Biotechnology, and almost all of you recognize James Watson here. I could put that here with John Dowling or not yet Chris, but maybe a few others, myself maybe, with a lot of hair and no hair, which I thought maybe I'll do in my next presentation. This was the first one, so I was in a hurry to prepare. But as you can see, there's a massive change that happens at the organismic level, tissue level, and same thing happens at the genetic level in genetics. The trouble in studying aging is that in addition to genetics or some genetic aspects of aging, there is a lot of stochasticness. So aging in general is believed to be stochastic process and adaptive process, that it sort of adjusts what an organism needs to do after its job is done, which is reproduction. And in many organisms, as probably all of you know, the organism dies after it has done its job or reproduced. So in humans, that is accompanied by this elaborate process of aging, which includes many specific changes, but there are many other more established, you can say, universal changes that happen in individuals, in different species, in tissues, cell types, or whatever. The commonalities that exist in these processes indicate that there must be some common pathways. There must be some sort of genetic control of aging. Something must be similar. That's why this is happening. But aging has been a subject which is extremely challenging to study. And the primary reason for that is really that these changes not only could be very specific, but their magnitude is generally very subtle. Now, it's sort of not hard to appreciate that if you have a disease or if you knock out a gene or something major happens, you're going to have big changes, both in gene expression, protein function, or cellular pathways. But aging is not that way. In aging, you have very subtle changes, but significant changes. What that means is that you cannot validate some of those changes so easily. It's an iterative process. You have to replicate some of that work many, many times in order for you to be confident about what you're saying. So therefore, data analysis and validation require a very different kind of approach. Aging has been studied by huge number of labs over the years, but sort of the data is just beginning to come together in some sort of pathways or in some coherent manner, I would say. And here, as you can see in this nice review from Anne Brunet, what she talks about are different changes that are happening in aging in different tissues. Much of these are fairly common changes that are observed in multiple different tissues and cell type, but I think they all in some way converge on this genomic dysregulation. So we'll talk about genomic dysregulation a little bit. So I have been interested in aging for a long time. After I got my tenure, I said, all right, now it's time to, there are two things then you start doing. One is study aging, and the other is look at evolution. And evolution actually is another topic that has become that is becoming a major part of my lab, and that story is still developing. So maybe in the next few months or a year, I think you'll be seeing some stuff on evolution. But aging is finally coming to an age, I would say. My first paper on aging was just when these microarrays were beginning to come up. We still did not have all these fancy methodology of today, and the microarrays were made on slides. This particular slide that was used in first ever experiment, which I would say probably in the eye field at least, but it was very, very, very early days. This slide has got few hundred or so genes that were spotted. We used to make our own arrays in the lab as well, but this was purchased. And by looking at that, we found at least 10 genes, and some of them we were able to replicate by RT-PCR that showed changes, very specific changes in gene regulation. And we used obviously retina at that time. But as I began to study further, it was very hard to figure out. We had another couple of papers on SAMP mice. Some of you know what those are. Those are senescence accelerated, aging mice. And in those mice, again, the retina ages like other tissues very, very quickly. But when we begin to look at the whole retina data, it was not very easy to make much sense out of it. And we sort of decided to then start focusing to our favorite cell type, which was photoreceptors. And within photoreceptors, we thought of rods because of multiple reasons. There were many, many papers in literature listed here. And you are recording, some of you guys can go back and read. I don't want to read what is written here, but the bottom line is rods are the first cells, first type of photoreceptor that seem to sort of lose their function, and they seem to die in normal aging and also actually even in age-related macular degeneration. And there's a lot of work done in early days by a lot of people. I listed only a few of them. But what was very interesting for me to see was that rods were also thought to be the primary drivers of aging-related synaptic remodeling, which I thought was kind of nice. People tend to ignore rods and think about only cones or inner retina neurons or ganglion cells. And being a photoreceptor chauvinist, here I am trying to convince you that rods are probably the most important cells in the retina. Anyway, we sort of begin to do a little bit more studies of our own, and this is from one of the papers that was published in 2010 in PLOS One. This time, we purified rod photoreceptors from NRL-GFP mouse at different ages and did microarrays. Some of you young folks may not know what microarrays were or what they are, but in early days, as I said, we used slide microarrays. We used to spot our own genes on these arrays, but we also were using microarrays. And also then there were Affymatrix arrays that had come as well. So in any case, we did some of these early studies and we found a bunch of genes that go up or down, but sort of nothing very satisfying, nothing that sort of was beginning to really tell us conceptually what is going on in aging photoreceptors and why there's a decline in function. And this very small decline is really not ONL thickness in ventral side, as you can see, is somewhat down, but very significantly down. And there is some change in scotopic A and B waves as well. So B wave particularly is very relatively easy to see the change with age. And so we were not very happy, let's put it that way. And I was just trying to figure out how to think about that. And around the time, many other people were looking at aging, and we thought, all right, let us think about epigenome, because these gene expression changes may not actually lead to proteomic changes or may lead to some other thing. But what is the driver of all that? What's going on? One of the things that was coming out in this decade, past decade, around 2010 to 2016, 17, was this DNA methylation was coming of age, particularly because we were able to use next generation sequencing technology, and we could look at DNA methylation, which is sort of a major kind of epigenomic change. All of you probably know that the major methylation in DNA is in the CPG context, where 5-methylcytosine is over here. That is basically is the major one. But now there are so many others, hydroxymethylations and methylations and other residues that are observed in the mammalian or in vertebrate genome actually in general. And this, again, these DNA methylation changes is also a little bit tricky to study during age, because with age, you see some errors in the epigenome, or what we call epigenetic drift. And I'm not going to elaborate on that, but what was coming out is that there may be some CPG islands, if you focus on that, those methylation and those CPG regions could actually predict the chronological age. And that was the concept of epigenetic clock put forth by Steve Horvath. But the problem has always been that people looked at some of these things in the early days by RRBS, or representation method, looking at only small numbers, or many people were using arrays and all. The other problem is, which is what the small fraction of genome that was covered, people are not covering all the genome. Another problem, of course, is that these changes in DNA methylation with age are dynamic, and some of these could be genetically determined, whereas some others could be stochastic. And the worst part is these changes are not very large. So how do you look at that? And many people use the whole tissues, which is not the appropriate way to do it, because you have mixed cell populations, and every cell could have its own distinct change in the epigenome, and so you're not going to be able to figure out really, the noise might be too high to look for very specific changes in DNA methylation. So we set up this hypothesis that maybe DNA methylation changes that happen with age, they are going to impact metabolic pathways, which in turn are going to lead to declining rod function. And we begin to look at in a more systematic and orderly fashion, and much of the work in the next few slides was the work of a talented postdoc in the lab, Jimena Corso-Diaz, who actually just recently got K99, so I was pretty happy with that. So Jimena worked, collaborated with a couple of other colleagues in the lab for bioinformatic work, but also she took some of the data from published data from other labs as well. And fortunately, a lot of people have been using similar system with NRL GFP mice, where the promoter of NRL is driving GFP, and you could purify rod photoreceptors at different ages of differentiation, and also we could do that for age. And people have used it for a variety of things, but much of the work has been focused on early stages of development and maybe disease conditions. So we did ATAC-seq, we have RNA-seq data, and we did cut and run with some histone markers. And we combined all that data basically with the whole genome bisulfite sequencing that was done. So we did an integrated analysis. So I'm going to sort of go over that. And in order for us to make any sense of the data, rather than using just 2 time points, 3 months and 24 months for this analysis, we took intermediate steps of 12 and 18 months as well. My general belief is that in many of these experiments, if you just have 2 time points, it's very hard to sort of trust many times. And if you see a progressive change in either gene expression or chromatin structure or DNA methylation or whatever, then it's much, much easier for us to say that that change is real. But if you just have 2 time points, it's very hard to sort of say, yes, you have to do still validation, but sometimes not that simple. So when Jimena began to analyze this whole genome bisulfite sequencing data, it was very clear that genes that are expressed in the rod photoreceptor, they show near this is the transcription start site, they show low methylation. This is pretty well known. I mean, so we are finding what other people find, and that is a good thing. That means what we are doing is right. Then using different kind, using the hidden Markov model and other statistical tool, folks in the lab, the bioinformatics folks working with Jimena, what they looked at are two kinds of regions. UMR are unmethylated regions in the genome, and LMRs are low methylated regions, and these are very usual terms defined by other people. And what we found, like everybody else has found, that much of the unmethylated regions are associated with either the promoter regions or within the intron. Some of the intronic, these things could be connected to alternate promoters that are quite prevalent in genes expressed in the retina. Actually, surprisingly high number, and that's a different topic we can talk some other time. But these LMRs, or low methylated regions, are really interestingly not only in this intronic region, but also in the intergenic region. And that's good, because that means these things are likely associated with enhancer regions. So what Jimena also did was to combine this with ATAC-seq data. This ATAC-seq data was taken from Mu et al. Paper, which is from Jeremy Nathan's group. And this was the active histone chromatin mark, K27 acetylation. And in both cases, if you have random, you look at the counts, they are here, whereas in UMR and LMRs, you see enrichment. And that's, again, something very nice, but nothing unusual, but just to confirm what we are seeing. This is just from 3-month-old data. We are not really looking at aging at this stage. This is just a 3-month-old rod methylome, and that's what we are seeing. And as one would sort of, again, predict, what we are seeing is something here, that this is a gene, PD6A, expressed in the retina, or in the rod photoreceptor. PD6C is not expressed in rod photoreceptors, and everything, including the presence of UMRs and LMRs and methylation and chromatin, open chromatin regions, active marks, everything is very consistent. We just picked PD6A and C because they're same genes, sort of different genes, of course, but one is in rod, the other is in cones. So we just sort of said, all right, so this is, everything is looking good. So now we can go and look at what happens in aging. So when we do that, this is a Pearson correlation. As you can see, these are different samples at 3 months, 12 months, 18 months, and 24 months. And then you can see the samples correlate pretty reasonably well. And remember, this is 0.94 correlation. So that's the minimum that is being shown here. And then R equal to 1 is over here. And when you look at that, you are going to find what you call differentially methylated regions, or we have used this term to extensively in the paper and in this talk as well. And these DMR, the different methylated regions are DMRs, and age-related DMRs, what they are. And so you can either have a gain of methylation, this would be also DMR, or a loss of methylation. Okay, and if you look at that, this is one of the chaperone genes. But if you look at that, these are DMR of 3 months versus 24 months. For the sake of convenience, we just plotted 3 and 24, even though we had all the data at 12 and 18 months as well. You can see where they are being gained and where they are being lost. What is happening in gain and in loss. In both cases, you can see the enrichment of these genes, particularly in the intergenic or the promoter regions or intronic regions. So intronic regions are here, and you can see big change here, basically. And here you have in the CPG island, there is a big change in the loss of methylation. So there are lot more actually what we found were hypomethylated regions where with age methylation was lost rather than gained basically. So that's interesting at the genes that we are looking at. But you find with age both changes in methylation loss and gain. But what does that mean? How does it correlate to genes and what those genes are? So one of the things we begin to do before we go there is to figure out whether there is some sort of organization in these DMRs. So this is, you will not be able to, this is a Sarkos plot where what we have plotted is on all mouse chromosomes, okay? So this is from 1 through chromosome 19. On all mouse chromosomes, we plotted the density of DMRs. This is rods, but we also took the DMRs from cortex as well as from non-neurogenic cells in cortex. Okay. Now you can start looking at where there are certain kind of hotspots where you see a lot of DMRs in different. So you can make conclusions that there may be DMRs that are common whether you are a rod or a cortex, another neuron, or a non-neuronal cell within cortex. So there could be common DMRs in all of them. Then there could be DMRs that are clustered specifically in rods, or they may be clustered specifically in all neurons. So you can find any of those things. We found specifically a large number of clusters on mouse chromosome 10. And they were actually, if you see here, these clusters, 1, 2, 3, 4, 5, 6, 7, 8 clusters were there in rods on this chromosome 10. And they were defined based on specific distance within million base pair, how many you see and things like that, using some very specific statistical criteria. And these were largely present in rods, many of these clusters. So look at this particular cluster, which is only in rods. And what genes does it have? It has genes which are related to, it has FOXO3 and many genes that are related to what has been termed, at least in by one group, as longevity interactome. These genes have very interesting correlation for longevity, at least in model systems. Interesting. And on chromosome 4 I mentioned, because there's a cluster of DMRs on chromosome 4 that match also with mitochondrial genes. So many of these clusters were turning out to be sort of fascinating if you want to go one-on-one. We also found a cluster on NRL, actually, for that matter, on chromosome 14. It was not too many, but right where it was. And then there were clusters on very specific genes or region of genes which could have obvious implications for what is going on during aging. And so aging might be inducing changes in epigenome at those very specific time points. So,
can I ask a question?
Yes.
So, can you go back one slide? So, would you suggest that there is this FOXO3 makes some kind of changes more in the retina than in cortex? So, cortex doesn't have this.
That is the suggestion. Yes, it needs to be tested, but yes, that is the implication. The implication from this kind of study right from here is that because of this extensive cluster, you can see right here, not as much, but here just too much of methylation, hypo or hyper. And all the genes here are associated with, have been associated in some of the other longevity. So we predict that that's what the case is. But you have to validate it.
Thank you.
Okay. Yeah, please do not hesitate to, because I cannot see the chat because I'm sharing my screen and the whole my laptop screen is the slide. So please don't hesitate to stop me if you have any question. Any other question now that I'm... No? All right. So then we thought, okay, those clusters are fine, but what does it really mean? So can we correlate that to gene expression? And it has been shown earlier that many DNA methylation changes in CPG do not really correlate very well with gene expression. I mean, there have been people have been trying to look at that. But I think part of that is technical. I think, and I hate to brag about what we have done, but I think we are using pure cell type rods, and we don't have contamination of even cones in there because we don't see cone opsin expression even. And you saw PD6E data. So I think our data is not very noisy. And second thing is we are doing a time course of aging, so we can have better conclusion. And the most important thing probably is that we are doing whole genome bisulfite sequencing. And what that means is we are multiple animals. So it's not like we did 3 animals, minimum number required. So with whole genome bisulfite, we are scanning everything. So whatever significant stuff is going to come out, the data that is going to come out, will likely have a little bit more meaning because we are scanning and after corrections and everything, whatever significant data is going to come will have probably a little bit more meaning. That's what my thought process is at this stage. So we thought, all right, let's correlate these DMRs or differentially methylated regions to the data from the ATAC-seq data that was published by Mo et al. And from Jeremy's group. And then we also did, or Jimena, I don't do anything in the lab, so I should say, when I say we, much of that is, most of it at least is Jimena. So what she did was this cut and run, which is a new wonderful technique that has come up. And what she did was K27 acetylation, which is an active mark, and K27 trimethylation, which is a repressor mark. And she plotted everything. And the way these plots are is they're centered at transcription start site in the genome, and you're looking at 0.5 kb on either side. And if you have something very red or yellow that are low versus high methylation and things like that. Okay, and so if you see here in the intragenic region and some of the other regions, you see a lot of clusters, and many of these clusters are correlated to ATAC-seq peaks. Here, the blue or the green are basically regions where you have those peaks, and white is not. And now you can see at 2 months and 3 months, whatever you're seeing here correlates very well with this. This is 24-month, how some of these changes might be happening in time. So this is CPG loss in the intragenic and intergenic region, and this is CPG gain, methylated CPG gain. So when you, I mean, this is hard to follow this way, but you can go gene by gene and then look at it. And in many cases, what we found was that these DMRs were actually localized in gene bodies. And within gene body, they were in the regions at the boundaries of open chromatin. So here is the ATAC-seq. This is a gene OSBPL8. And this is the start site, of course. This is within the gene body. There's a huge chromatin region, open chromatin region, suggesting that this is actively transcribed in the rod photoreceptors. And you can see the change that is happening in the CPG here. There's an increase here. And here, again, in 24 months, there's an increase in methylation. You can see it at the boundary of this for LiPo1 gene. So basically, you can start seeing, there are many other things that have come out, but I'm not going to focus on that at this time. But look at these transcription factors here. there is a loss and gain, but sometimes you'll find a gene where there's both loss and gain in the same gene, but in different places. And the implications of that have to be validated. I mean, you have to look at not only gene expression and how eventually those changes are impacting gene expression, but you also need to look at you also need to validate some of those things. These are all predictive. These are all suggestive that these are different transcription factors, and CPG loss is of course much more prevalent than CPG gain in many of these transcription factors that are listed here. So now you come to genes. Okay, so you have these DMRs and you have this open chromatin region, and what are the genes in the neighborhood? What genes? You can correlate that with the genes or transcription start sites where they are, or if they are intragenic, it makes it very, very easy. Intergenics are a lot more difficult to sort out. So you can focus on some, and this was unsupervised clustering that was done. And in this unsupervised clustering, you can have different kind of clusters. This is an early cluster that is from 3 months to 12 months. There was this change. These are z-scores, so it's easy to plot because you are normalizing everything here. So this is just right in the beginning, it goes down, and this right in the beginning goes up, and these are more progressive change, and these are late changes. They happen only after 18 to 24 months. And you can look at the kind of genes and pathways. Look at AKT3 where it is coming, and there are a bunch of other interesting genes, but we did not care too much about many of these pathways. Okay, why? We wanted to see what happens to metabolic pathways or mitochondria specifically. Why did he want to do that? Based on this analysis. This was done by Anupam Mundal, another talented bioinformatic postdoc, and James Gentry, who is a postback. After 2 years, he will have 2 first author papers in major journals. We are submitting it, and he's going to go to graduate school in Virginia to do bioinformatics. So we'll have a hopefully talented person who will stick around and help in computational biology aspects in the eye field. But this integrated network, actually, what it did was it took all DMRs, and it also looked at RNA-seq data. So it looked at methylation data, RNA-seq data, but also it could have sort of converted those genes to proteins and build the protein interaction networks, which are directly correlated. First order protein interaction. We did not want to go to second order or third order, just first order protein interaction. And you find with these that there are 3,958 proteins which incorporate 622 DMRs. I told you, right, I probably didn't mention that we found about 2,000 or so DMRs which were changing. They were present in aging rod photoreceptors. And by doing all kind of statistical analysis, what was thought to be significant was this ratio of, where is mine? Okay, 0.08. And then you can look at downregulated genes, upregulated genes. This is very hard to figure out, right? It's pretty sort of dense and all that. So I'm going to go further. I'm going to show you a little bit more cleaner version. This is DMRs that we picked up by using KEGG and reactome pathways, and they were plotted, and these are all FTR of less than or equal to 0.1, which is fairly significant. When you start looking at that, and we plotted where at least there were 2 DMRs in gene associated with the pathway, rather with a pathway. You start looking at that, and you start finding many pathways that are known to be associated with aging, but not in this way. People have, based on gene expression profile or all that, people have predicted that. But we showed that in aging there's a change in epigenome, and that seems to be affecting these very specific pathways. Some of these are universal, and some of these are rod specific changes that are happening. But still, it is not very easy to decipher, but we'll focus on this part maybe. We'll focus on metabolic pathways. So this is a subnetwork. Now you can start going deeper and deeper into this network and start focusing on the genes or proteins that are associated with that. Look at what is coming out here. People who are mitochondria aficionado will quickly realize that there's a just huge network here. And in RNA-seq, there are down-regulated genes, up-regulated genes. These are down-regulated genes. Some of these are up-regulated genes. And then you also have changes that are coming late, and then changes that are coming early in triangle. Look at hexokinase here too. So, I mean, you have all sorts of, and then there are progressive changes and things that are in line, any circle. So things that are not having any line around the circle or square, that means there's no DMR, but all of these have DMR, but differentially methylated regions. And that was fascinating for us because suddenly we figured out that there's at least one pathway that has been proposed to be associated with aging in general. We are able to sort out based on epigenomic changes in rod photoreceptor. There are more, so don't get me wrong. I just sort of, if you go here or here, you can pull out many other pathways. Okay, and some of these are related to synaptic remodeling, which I briefly mentioned in one place, and some of these are related to proteostasis, which I think is another very important pathway that is related to aging. But some of the changes in these pathways are rod specific, where other changes in those genes are much more universal. So you can see this is just a pattern of based on the genes that we were showing, the changes in gene expression. This is a log 2 fold change, so you can see. Now these changes are not all or none. You're not going to have some part of mitochondrial protein disappear with age. You're not going to see 10-fold change in any one of these things. But there is a sufficient change, log 2 change, 2, 4, 6 log 2s. Okay, based on that, in some cases, there's a pretty significant, these are all significant, by the way, statistically, in all of these cases. So we said, all right, we do see some changes. We see some changes in mitochondria, some changes in metabolism, some changes in fatty acid but can we validate it? Can we do something more? To do that, a few years ago, another talented postbac, Keshav Koragwala, and working with few others, had optimized this very nice method. And we took a lot of help in this from our colleagues in NHLBI who work on heart mitochondria, Dr. Bob Balaban and his colleague Rahul Kobian, because they didn't believe seahorse. They said, you can't put rotinone this way or that way. They had their own ideas. So we worked with them and finally convinced that the way we are doing is correct. And I don't want to go over that part. But what I want to tell you is here, rather than using cells, we can take that whole retina and put it in, dissect it, and put it in tiny punches of equal size, and 1 millimeter or 1.5 millimeter or whatever, and we put it on a mesh, and then we have these microplates, and we can secure that and use these microplates in seahorse XF analyzer. And we have done tons of experiments, lot of optimizations and everything. And based on that, we had shown earlier that retina, particularly photoreceptors, and because photoreceptors in adult retina are more than 70-75% of cells, Also, if you take degenerating retina, it doesn't have photoreceptors, then it's different. That reservoir capacity in these mitochondria is very low. And that is because photoreceptors are very efficient in using the oxygen and coupling it to generate ATP. And that, in general, you can, when you add an uncoupler, you can double the oxygen consumption in many cases, in most different tissues, and including in degenerated retina where there are no photoreceptor. If you take, you'll see a major twofold change, but it will be much smaller change, 20-25% in photoreceptors. So another postdoc in the lab, Coco Jian or K Jian, she worked with Jimena and tried to figure out what is going on. So here's the hypothesis. Let me explain that. In retina, mitochondria primarily uses glucose as a substrate. And these are some of the genes that are not only having DMRs, but their expression is going down. And this is the glycolytic pathway which generates pyruvate, which goes into TCA cycle and out phos. And then this comes from beta oxidation, and this comes from fatty acid, and some of these genes actually were increasing, suggesting a hypothesis that maybe with age, glucose is being utilized a little bit less. It's not all or none kind of phenomena. It's just that fatty acids, which are generally not used in the young animals, in young retina, now are being used much more in this. That's the hypothesis to that. And we, of course, use rotinone to inhibit complex 1 to see everything what is non-mitochondrial respiration. And we use BAM15, which we think is a much better uncoupler than FCCP, and we presented evidence in previous papers. So this is an uncoupler which tells you the maximum respiration that is there. And we normalize that OCR or oxygen consumption rate with cytochrome A, because you want to make sure that the number of mitochondria is not changing. Okay, the amount of mitochondria is not changing. So you do that, and when you look at that at 3 months and 12 months, no, 3 months is here, at 12 months and 24 months, there's a decrease in OCR. Pretty significant decrease, 20%. But that's what you have, 15-20% decrease. But the maximum respiration does not seem to change. What that means is the reserve capacity, which is calculated based on maximum respiration and the basal respiration, which is here, so much less. In 3-month-old is about 15% in this experiment. Here, in 24, is almost double. So mitochondrial reserve capacity now is increasing. That means mitochondria are not efficiently using the oxygen for ATP, as well as they are using less oxygen anyway. So what does that mean for our hypothesis? Okay, then we wanted to see what happens. Is this really true? This is all done with glucose as a substrate. So then we looked at fatty acid dependency using an inhibitor, etomoxil, which inhibits an enzyme called CPT1A or carnityl palmitoyl transferase, actually, which is very important for fatty acid oxidations and all. And if you take the young retina, if you put etomoxil, there's very little change. But now if you do this is a kit that is available, and if you now take in old retina, you add etomoxil, and now there's a much larger decrease here. If you quantify that, you can see that in young, there is much less, but with age, and this is the amount of etomoxil, by the way. With age, a 20-month-old retina shows much more significant amount of consumption of fatty acids than it was earlier. So if you look at these things. So what that suggests to us is that there are definite changes that are happening in energy homeostasis. So to conclude now, I would say DNA methylation changes occur with age, and they are in regulatory regions in rod photoreceptor genes. And genes expressed in rod photoreceptors, they're not specific genes only, but they're specific, and other genes that are expressed in rods. These changes that are associated with age, they cluster in specific chromosomal region. They associate with gene expression changes in cell type specific and shared pathways. And you can decipher some pathways that are affected by aging by combining these different data sets. We focused only on one. Remember, in this particular one, we only tried to look at mitochondria. We didn't talk anything about the synaptic stuff, or we didn't talk about proteostasis. We don't talk about other metabolic profiles as well. That is yet to be done. And we also need to go to proteomic studies, particularly in rod photoreceptors, to make sure to get much more deeply into the mechanistic aspects of these changes. Because some changes in gene expression, specifically in mitochondria, may or may not be correlated to proteomic changes. So but at least we were able to validate to some extent what is going on. These changes are not what you generally expect to see or in knockout models or when there's degeneration or whatever. But these are all highly significant and have been repeated, replicated, reproduced by different people. And so, but they are what they are. They are 15-20% changes. And remember, that's what change you get in rod function as well, in physiology. So based on all that, this was something that I had proposed in one of the reviews in Progress and Retinal and Eye Research a few years ago, that when we get old, and this is based on not our data, this was based on a lot of work that is being done by lots of people. We are just putting it in our context, putting it for our work. And I don't want to take any credit from that, but we synthesized that to put together what is going on as we age. You have tons of these environmental influences: light, hypoxia, stress, different kind of damages that accumulate in the retina. And in photoreceptors, these are sort of crying. These are tears of these photoreceptors. And what is happening is that we predicted that time based on other works that mitochondrial will be one part, other will be proteostasis, third could be epigenome, but there could be many, many more changes that may be happening. And the adaptive response is decreasing with age. So it's a sort of yin and yang, a balance of at young age we are able to take care of everything. Now if you combine this with genetics, then it makes it even more fascinating and interesting with respect to disease process, that what is going to lead to if this balance, if things go off balance. So again, several years ago, I was telling you, I've been thinking about aging for a long time. So in 2009, when I wrote a review in Annual Reviews of Genomics and Human Genetics, we were just trying to sort of say how with age, early AMD and late AMD are changing. If you have physiological aging, this is at this rate, but at genetic and environmental factor accelerate the onset and severity. So we were thinking in a different way. We were not saying that genetics is the driver or environment is the driver. I've been trying to say that age is the driver for disease. What genetic susceptibility and environment do is accelerate the process. but they can also slow down the process too. They can also reduce the severity. So just imagine you are beginning to see some of these epigenomic changes with age. Can we somehow reverse it back? Is it possible that the ARETS study with antioxidants and all the other wonderful Mediterranean diet that Emily Chu and other colleagues have been wondering, these environmental factors, maybe they are preventing the epigenome to age as rapidly? Could that be a prevention? Could that be the cure? More than that. So I think that's what I have been thinking, that I've been talking about. So I would sort of argue with you. I would suggest that something that we have been ignoring for a long time, I think, which is aging, is finally coming of age. Pun intended, of course, here. Genetics and environment are there to either accelerate or decelerate the onset of a disease and its severity. So the way I like to put it is, I'd like to put aging on top of this pyramid. And I would like to say genetic susceptibility, environment, diet, they all in some way impact epigenome. And that epigenome change can be altered. Impact of aging probably can be altered by environment, diet, smoking, or by genetic susceptibility, and vice versa. So we need to look at all of these things in the context of epigenome. We need to look at all of these things, how as an organism we and our different tissues and cell types are responding. And I think maybe there is a way.
Anand, can I just ask you before you go?
Sure.
Because I see that you're going towards, did you look at the senescence pathways? Because it's very fascinating whether
Yes,
the photoreceptors senesce or not.
We did, but then we didn't focus on that. There are some senescence accelerated secretome proteins that are also part of this.
Okay. All right. So, yeah.
Another quick question here, Karine from Allergan.
Yeah.
AbbVie. Were you able to pinpoint any of these DMR changes to specific diseases of broad diseases or any retinal diseases that one can pinpoint and try to change?
I said that right in the beginning, that aging is going to be it's a low level subtle change in many places. Now you have to have additional susceptibility in order to cause the disease. We specifically did not focus on any one disease at this stage. We intend to, but I think over the last few years, so maybe I'll go back and tell you my mindset. So when I was young, and now I'm aged, when I was young, I used to I love genes, and I still love genes. That's why I changed from all the biochemistry beautiful that I'm coming back to work on genes. But then I have now discovered pathways. I have now discovered this much more global stuff that I can focus on. So I no longer am trying to focus on any one gene, and I'm looking at how we can target specific pathways. For example, I don't know what you were trying to get at, but if you are looking at, say, AMD, and if you say, is there anything on CFH? Okay, so if I find something at CFH, what would that mean? You can have maybe a different kind of methylation on CFB, on something else, on another complement pathway gene, CFD or something. So I think we need to take it in total, we need to put that in context, specifically when we think of AMD. Something else that, so I really am not very interested in, people have been trying very hard to target one gene for AMD or one even pathway. We are yet to succeed in that area based on just that. I think we may need to target multiple pathways. I think we may need to target multiple genes if we want to cure a disease like AMD. But I'm suggesting even something different. I'm saying that if you could change the underlying epigenome that is associated with aging in some way, maybe by diet or maybe by some other means. After all, remember, caloric restriction has been associated with prolonged life. So if we could do that, wouldn't it be fun? Wouldn't it be? So I'm sort of now thinking, rather than thinking about genes, I'm thinking about more what comes after genes. I'm thinking more not transcription, but post-transcriptional changes. I'm going more and more into thinking about transcription termination, transcription elongation, transcriptional or translation and post-translational. So to me, the epigenome, whether epigenome is at the DNA level, which we talked about today, but another interest of mine, more recent research, is the RNA epigenome or epitranscriptome, what is being called, because RNA has tons of modification. What are those affecting? So to me, those changes are going to be pretty fascinating and critical, and can we change them in very targeted manner by drugs, by diet, by changing different conditions? How do we go there? So to answer your question, no, we have not looked at any specific genes, and I don't intend to, because I am not as keen. But all the data is out there, and all the data is freely available, and even the basic files of every kind. And we're happy to give it for free to everyone. Whatever anybody needs to look at.
I have two questions. So hopefully, I'm sorry, Corinne, you wanted to continue?
No, just a quick follow-up to this question. Do you think, let's say even we pinpoint a certain gene, would we be able to change, to target methylate one gene up or down? Or, I find it difficult in my mind to
yes,
see how we'll be able to manipulate methylation in one specific gene or one.
I fully agree with you. I think it'll be very hard to target one gene, but there are ways to do it. It's not going to be easy. That's a long discussion. I can, maybe you and I can do that if you want to do a single gene in a very specific way. But in my own lab, we are not trying to do that. Okay. In my own lab, we are not focusing on one gene. But if you want to do it, it can be done. It can be done by very simple way. Could be CRISPR gene editing as well. So you might be able to target those things, and you can do that. So, but there may be other ways too.
Thank you.
This is Chris Kenny. If I can ask you something,
Sure,
have you looked at the mitochondrial DNA and the sequencing of it as it might correlate with some of the changes? And the reason I ask is when we look at the human and we look at the mitochondrial DNA, and we use our hybrid model where all the nuclei are the same, but the mitochondria are from different groups of people, let's say maternal African or European or Asian, and we put them in our hybrid cells, the epigenetic, the total global methylation patterns are different, the expression of epigenetic genes are different, and so the DNA within the mitochondrial DNA affects the epigenetic nuclear genes. So have you ever looked at those, let's say the haplogroups for the mitochondrial DNA or the heteroplasmies, the increase of number of heteroplasmies in the mitochondrial DNA?
No, we have not looked at that with age. These are mice, and
okay.
So in our genetics work, in AMD genetics work, I've sort of, I'm very familiar with what you and Emily and others and have done, and these papers. We sort of didn't want to focus on that because to me it looks a little problematic because of heteroplasmy. So that's there's just so much going on there. Can I make a consistent change based on that? But the way you guys are doing based on haplogroup is very interesting. And I personally believe, based on some of the other work which has just come out on stem cells with a colleague of ours, where there was clear mitonuclear signaling and some other work that we have just submitted. I think the mitochondrial proteins and mitochondrial DNA, mitochondrial proteome, actually, I would say, is even more fascinating. Mitochondria, 15% of mitochondrial protein have not been characterized yet, and we just submitted a paper where we show function of a uncharacterized gene, which is evolutionarily conserved right from gamma proteobacteria, and it has a D-glycase function which cleans up mitochondrial proteins as they're being synthesized. And if you don't have this gene, you have cardiac fibrosis, you have age, and these end products, advanced glycation end products everywhere, and there are all sorts of things happening. So yes, mitochondrial proteome and DNA has to be maintained pretty well. And I think mitochondria will be talking to and actually dictating to nucleus what to do probably, and we have not understood some of that at this stage.
Okay, Anand, since everybody was nice to you, maybe I would just ask you a few questions because I totally disagree with you. So I think that would be the point of initial question. Would it be easier and maybe more informative to focus not on aging diseases, but like RP disease, that you have in young organisms, whether it's mice or humans, they can function relatively properly. And only at certain age, they switch and they go through this massive degeneration. So the element of timing is taken off. The second thing is our life, it appears to be a program. And our hormonal, for example, as we pointed out, our hormonal composition over age, it changes dramatically. And so all of those epigenetic changes could be consequences of already programmed aging that we're facing.
I don't disagree with you at all. I think what you're saying, but it no way contradicts what I'm saying as well. What I would add to what you said in response to your second comment, I would say it's not a question really, it's a comment that because of changes that are happening in our genetically programmed part of aging, there are a lot of what we call stochastic changes that happen with age because of our and those aging associated changes in particularly in epigenome that you're talking about, they are not consistent in everyone, and every human being and every person has got very specific kind of there will be a lot of things shared, but a lot of will be different. We all, after all, have millions of genetic variants, which is what actually to some extent leads to epigenetic drift as well, right? So I think what you're saying is completely in tune with what I am saying. All I am saying is that would it be possible for us to change that epigenomic, to reverse that epigenome change, whether it is genetically programmed or coming from environment or from anything else, by doing some specific targeted therapy? I mean, after all, that therapy could be based on CRISPR, or could be based on diet, could be based on genes, could be based on pathway or small molecule drugs that focus on specific pathways. Absolutely. I
just wanted to rattle you a little bit. Now, I know you agree, and I think we're thinking alike on this. Anybody else? Let's have two last questions.
But this is not the end of the talk, I hope. But I have a actually question about you touched upon something very interesting about the stochastic versus programmed aging. So I will ask you, did you try to look at the methylation clock in your photoreceptors?
Yes, we did.
And
yeah, we did. So in the mouse, the methylation clock is not. Okay,
it's
so
a little bit, yeah.
You are taking me to a different direction, but I'm going to comment on that.
Just comment.
So the mouse one is we commented on our in our paper very clearly. We use the methods and it doesn't work, at least in our context. In human, we have done huge amount of work on epigenetic clock, and we are finally after 3-4 years worth of work, we talked to Steve Horvath pretty often in the beginning, and we have done these huge methylation studies in human. And based on all of that stuff, what we found was that the clock that he has does not work for neuronal tissues. So what we have done, what clock means based on the methylation CPG islands that he has, but now we have designed our own new clock based on our new data and the data from other neurons, and we have now a neuronal clock which functions pretty well. So we are finishing that paper, and with tons of human methylation data and how we made the clock by using this. And
okay. Fantastic.
Okay, let's go with final question to Daniel. Daniel, are you there?
Yes, I think before you go there, let me just sort of at least acknowledge the people we want. So this is Jimena, and this is Anupam, who did a lot of work. James, his picture I don't have. Ryan is now doing MD PhD at Michigan. Coco and Jacob, both our mitochondria folks, and Rahul is our wonderful collaborator from NHLBI. He is with the staff scientist with Bob Balaban, and this is our lab.
Hi, now this is
postdoc position if somebody wants to apply to work on this exciting stuff.
Anand, hi, this is Dan Chow from UCSD. I really enjoyed your talk. So I had a question, I think you hinted on it, and I was wondering, how much are these epigenetic changes conserved in human aging in photoreceptors, and have you done experiments related to that? And conceptually, would you think that there would be some differences based on the kind of unique configuration of the retina in humans versus mice?
I think that's a great point. The trouble is we do not have as much or as many human samples from younger individuals to really accurately answer your question. But most of our samples came from Deb Farrington, and she has some some samples that were more like 50, 60-year-old folks, and there was a bunch of others very, very old as well. So we have looked at that, and we have that's a different way with MQTLs, and we are combining that with all that, and we are checking it with the mouse stuff. So I think you probably should wait for about 3 months. Hopefully within 3 months we should be submitting the paper.
Okay, and as I expected, it was so wonderful. Thank you very much. And
I thought, Chris, you'll be interested in degeneration part, but I thought, all right, I thought you're done already. Okay, I'm sorry. I'm just joking with you.
All right, so why don't you stay and we continue?
Okay, thanks.
Thank you, everybody.
Okay, bye.